Okay, so today we have, for the graduate session, we've got a little bit of a grab bag of topics. So the first one is this idea of how do we transform from a map of saccades to the actual structures that generate the saccades, because the coordinates of them are different. Like the saccade map is, saccade vectors at each point, same amount of activity, saccade vector at each point, and then that gets turned into something that actually causes varying amounts of activity in the burst neurons that make the different size saccades. So we'll talk about that, but then there's another that one's straightforward. There's another more subtle one, is how do we do the inverse of that? How do we take some firing rate of eye position and turn it back into a location on the colliculus? That's a more subtle problem, hasn't been talked about. It's known to occur because we know that this remapping phenomena occurs, but it's less clear how it actually works, but we'll talk about what's known. And then the second thing is target choice. So what happens when you're looking out at the world is a bunch of junk out there. You got to decide to look at just one thing. If there's two interesting things, you don't want to look in between the two interesting things. You want to pick one and look at one and then look at the other one. So WTA is winner take all. So how does that work? And then finally we'll talk about a human VIP. So I'll talk about some experiments trying to find out where the human equivalent of VIP, because the experiments I talked about in VIP were originally done in macaque monkeys. Okay, so let's start off with the spatio-temporal transformation, or instead of convert, we'll call it transformations. And so why do we call it that? So we call it that because if we have the colliculus, so here's the left colliculus, and here's the, make it plenty big enough, right colliculus. So this is right superior colliculus. That makes saccades that go into the left hemifield because it's past the pons midbrain junction. This is the left superior colliculus over here. And so what we said is there's this map of saccade vectors in the colliculus, a motor map in the deeper layers of the colliculus, and that underlies a retinal map, and those two maps are lined up so that if you get some activity in the superficial layer at a particular point in space, if you go down to the deep layers at that point in the colliculus will cause a saccade to happen that takes the center of gaze to wherever that stimulus is. And so the idea is it's set up so that if a target appears in the superficial colliculus, you get the correct saccade to go to it. So, but the problem is the amount of activity that you have in a particular part of the colliculus is the same for small saccades and big saccades. So if you look at how much activity comes out of the colliculus for a small saccade part, it's the same as the amount of activity that comes out of the colliculus for a large saccade part. And if you stimulate with the same amount of stimulus in the small saccade, you get a small saccade. If you use the same amount of stimulus in a large saccade, you get a large saccade. So it's coding things by the location of the neuron and not by the amount of activity. Now that's different if you go down to so here's we'll put the pons midbrain junction down here. So this is pons midbrain because some of these things are behind it. So if we look at one of these neurons, say we take let's go down to the brain stem and talk about the structures down there. So we had those funny structures for horizontal and vertical. So the the horizontal one, we'll put the horizontal one here. So that was the P, R, F, horizontal. And that guy then projects out to oculomotor, oculomote, and oculomotor goes out to the muscle. And so this is my drawing of the muscle. So connection from these burst generators that generate the burst to make the saccades, and then these guys go out and synapse on the muscle. So if we look at this horizontal burst generating structure, if you have a big saccade, it makes a bigger burst. Doesn't care about vertical, so I won't do anything to vertical, but the size of the horizontal saccade corresponds to how big the burst was. So that's why it's called temporal. So the spatial mapping is like it's where we are in the colliculus. The temporal mapping is how much firing are you doing, and how do we convert between those. And then remember there was another sort of vertical structure. So we'll put the vertical structure up here. And that one had this funny name, the rostral interstitial nucleus of the medial longitudinal fasciculus, but we just know it as vertical. And so that one also projects to the oculomotor nuclei. So that's going to go to the oculomotor nuclei also to make the saccade, the vertical component of the saccade. So what's one way that we could do that? Well, one straightforward way is if we have, say we have a saccade that has like a bigger vertical and a smaller horizontal. So that when that deep superior colliculus cell comes out, so it's got a bigger vertical, smaller horizontal. So it's going to go down to the vertical and make a big synapse, a lot of synapses, and then it will go down to the horizontal and just make a small amount of synapses. So we'll make multiple synapses. That's a little clearer. So then if we go to a place that has, say, a big horizontal, but not much of a vertical, say this was like, down vertical and up vertical, so now we're like about the horizontal. So then what's going to happen is we'll go to the vertical and just make a small synapse there, and then go to the horizontal and make a lot of synapses. So that's this very straightforward way. There's some evidence for this, where you could essentially turn a code that's in the form of the same amount of activity but in different places into a code that is the amount of activity, and then break it up into the x and y, or the plus x component, the plus y component, and the minus y component, because you need a muscle to sort of like pull the eye up, and you also need a muscle to pull the eye down. But the muscle that pulls the eye outward, there's only one of those. There's only one that we're interested from the point of view of saccades. So that's one way that we could do the spatial, spatio-temporal transformation. But now there's a problem, because we said that we know that there is some method of monitoring eye position. I got to move my midline over a little bit to make some space. So we know that there's a method of monitoring like what the spatial location is, and how does that work? That works. There's this structure called, we'll just call it pH for prepositus hypoglossii. but that was basically the eye position. And how does that one work? That one works, neurons that are sort of sitting inside the oculomotor nuclei sample the final answer and send it back into prepositus. And then prepositus has to somehow get rid of the burst part, because remember the cell goes like, if this is a graph of the cell, it makes a big burst to sort of move, and then it has a continuous part, and then if it moves the opposite way, it sort of makes a little inverse burst, and then goes back to the holding position. So what we need to do, prepositus hypoglossii somehow has to get rid of the oculomotor neurons have that burst, but you have to get rid of that burst. And then this thing is basically the eye position. So it just needs to get the eye position out. And so we know that projects up to the, the we'll call it the midbrain tegmentum, or profundus mesencephaly. Midbrain tegmentum. I'm not going to add too many more parts. This is about it. This is profundus mesencephaly. So that's the deep in the midbrain. Okay, so we know that prepositus goes up there. But then it goes into the colliculus. And now we've got a problem. How do we actually turn varying amounts of activity here that are signaling eye position into a location? Now, we know that the neurons in the profundus mesencephaly go up into the colliculus, and they cross the colliculus, they actually go into the opposite colliculus, like that. And they branch like this and make these lines across the colliculus. So that much is known. But how is that going to help us? So anatomical studies done many years ago by Dennis Dacey in the snakes have shown that in the snake superior colliculus have shown that there are, and this has more recently been shown to occur in mammals, that if you look in the colliculus, you can find neurons in some layers of the colliculus that have an axon that goes off to one side and makes synapses, sort of a displaced set of synapses like that. Now, nobody's recorded from these. We don't know, whether this is how it works. So what I'm telling you is just like an idea for how it could work, or it's an idea for an experiment. It's not even an experiment. But we know that these cells exist in the colliculus, and they could be doing something else. They could be doing something to do with direction selectivity, for instance. But let's assume, for the purpose of argument, that we synapse on one of those cells, and let's say one of those cells has a little sort of offset in it where it projects sideways in the colliculus. And if we imagine that, all these profundus mesencephaly cells are synapsing on these guys at all different places in the colliculus, and they're all sort of pushing left. They're all sort of like sending a signal slightly off to the left. That might be one way that we could essentially shift some activity, sort of move some activity in the colliculus. And so if we just leave them on for a while, they could sort of like, continually move activity. They also have to be able to move it into the opposite colliculus because we know that if you need to make two saccades and the targets, say you had two targets in the colliculus and you saccade to one of them, it's possible that it would cause the other target to go into the opposite colliculus. And so we need to be able to move things between the colliculi also. And we know that these axons from the midbrain come and go into both colliculi. So that might be a way of kind of moving stuff in the colliculus. It's not enough because we need some method of moving things in the other axis. And we know that there's a structure that gets input from the oculomotor nuclei that has vertical eye position information in it. And that one is called, it has an even more ridiculous name, that's called the interstitial nucleus of Cajal, Ramon Cajal. Interstitial nucleus of Cajal. So that one has, so this guy down here has horizontal eye position, the prepositus hypoglossii, and this guy, it's not a connection, so it's a diagram label. This guy has vertical eye position. And so this guy gets input from the oculomotor nuclei that are sampling what was the final answer output for the vertical. So this is from vertical oculomotor. That's in a different place than horizontal oculomotor. It's in the midbrain. So vertical oculomotor feeds back, and nobody has seen these yet. But but what if we had some cells that either coming from here or coming from the area right around here that went into the colliculus and went across the colliculus in this direction, like that. And so those cells would then need to synapse onto cells that had a projection that went either up or down in the colliculus. And we don't have strong evidence that those things exist in the other axis. We don't have evidence that they don't exist. So this is probably, it's not very well worked out, but this is probably the best worked out piece of information. We absolutely know that we can shift targets in the colliculus. That we know. But we can shift them in the X and Y direction. And we know that the information for doing that from physiology experiments, we know it's coming from down here or from the interstitial nucleus of Cajal, these things that have eye position signals in them. So we know that these two structures, prepositus hypoglossy, interstitial nucleus of Cajal, project into the colliculus, and we know that they contain eye position information, and we know that they're responsible for moving targets around in the colliculus. So we know that we know that the moving is occurring. Exactly how it's occurring is not clear. But what's the bottom line here? The bottom line here is we need to be able to convert between some kind of map of saccades that's lined up with the retinal map and these firing rates of the burst neurons that sort of program the size of the saccade. So we know we have to sort of go from same amount of activity at a position to different amounts of activity, depending on what the position was. But we also have to go back. We have to take that different amounts of activity, sort of in a Cartesian-like coordinate system, and then turn it back into a location in the colliculus. Now, this, at first you might think, well, this is kind of a very specific thing. Why are we talking so much about this? But think about any kind of motor movement. You've got the same fundamental problem. It doesn't look geometrically exactly the same, but you've got muscles and motor neurons that drive muscles, and those muscles cause, if you contract, the biceps muscle, it causes the arm to move in this direction. If you contract one of the shoulder muscles, it causes the arm to move in this direction. If you contract them both at the same time, you get some complicated endpoint of your hand. Now, you've got a visual representation of what's going on. that's a map. And so if you want to grab something, you're going to have to sort of, you've got an object there and you're aiming to grab it using these neurons that are like the ones in the burst neurons. They code for position of your hand in space by amount. And we've got the same problem where we're going to need to sample what the muscles did and feed that back into the system in order to sort of update targets that you might be reaching for. So this is a general sort of problem, the sort of spatio-temporal, temporo-spatial problem. It's a general problem for the motor system for, because muscles sort of pull in one direction, basically, and visual, auditory, somatosensory stuff comes in the form of a map. And so we need to be able to convert back and forth between a map and these muscle coordinates, and then go from muscle coordinates, back into a map. So it's a general problem, not very well understood at all how this works, but probably the superior colliculus is the best worked out one. Okay, so that was the spatio-temporal, temporo-spatial transformation. So now let's talk about target choice. So one of the problems, obvious problems you have, is there's a bunch of junk out in the world. And we said that you have a saliency map in the colliculus in the intermediate layers that says, here's the location of things that are interesting to look at. And we also know that that saliency map can be updated. Every time you make a new saccade, you have to sort of shift that saliency map to take into consideration where your eyes went to, because it's basically in retinotopic coordinates and visual map coordinates. But now the problem is, what do we look at? Because there's several things out there. So we need some kind of system in order to pick one of the targets so that we can just move to one. We don't want to move to the average of two interesting targets that are, say, you got an interesting target to the left and the right, look in the middle. No, you want to look at sort of the left target and then later on look at the right target. So if we draw the colliculus again, so remember there's lots of cells in the colliculus. There's like 14 or 15 layers in the colliculus and maybe like 50 cell types in the colliculus. So we've got sort of a complicated thing that we're just sort of scratching the surface of. So here's all those layers in the colliculus. So we know that the retinal input comes into the top of the colliculus, to the top third of the colliculus. Then the intermediate layers are more where you have the saliency map, and the deep layers are where you have these neurons that just burst right before you're going to make a saccade. Those are the ones that I drew over here. So these, let's label those guys. So this one is a deep superior colliculus burster. That guy. Okay, so, but that's just one of the outputs, but there's, 50 to 70 things that are coming and going from the colliculus. And so let's just pick out two of the additional things that are coming from the colliculus. And these two are in the midbrain. So they're in the midbrain. And they've got strange names. We'll call, so let's make them down here. So everything's a box. So these, and sort of the generic name of this is the cholinergic nucleus isthmii. So what is isthmus? That just means kind of like a place on a landform that narrows down to a little sort of narrow passage. And what is the narrow passage? The narrow passage is, if you look at the midbrain, the superior colliculus is kind of bulbous. If you go behind the superior colliculus, it sort of necks in. So that's the isthmus of the midbrain. And there's a neuron, there's a structure in there in all mammals and vertebrates. And fish have one, and reptiles have one. It's been there a long time. It's called various things. So in reptiles, it's called, nucleus isthmii. In mammals, it's called parabigeminal nucleus. Say reptiles. And the terminology is not very consistent. So in reptiles and birds, there's several nucleus isthmi. We'll come back to that, but it's called the parabigeminal because it's got two little bumps, nucleus. Probably never heard of it. That's okay. So but it's cholinergic. You can tell that's the same thing from the neurotransmitter. And Cahal saw these neurons very early on, and if you look at these neurons, what they look like is so here's a neuron down here. There's a map. There's actually a visual map in nucleus isthmi. And where does the visual map come from? Well, it comes from neurons that project down to nucleus isthmi, and different neurons that are in different places project down to different parts of nucleus isthmi. So if you go into nucleus isthmi, you can find there's a clear retinal map in there. But then when you look at the projections that come back, these were recognized very early on. So they come back and they go back to the same place in the colliculus, and they make these really characteristic terminals with thousands of terminals on them. And if you go in the MRI center, I drew one from a turtle. And Cahal, so you can clearly see they're just nailing that part of the colliculus. Cahal called these terminals that look like paintbrushes or brooms, because they look like a little sort of wicker broom or a paintbrush. And so this guy will come out and synapse in a similar part of the colliculus over here. Now, when you look at this, you say, okay, well, it's good and all that, but that doesn't look awfully stable. And why isn't it stable? Well, this is an excitatory projection coming here, and we know that this one's an excitatory projection too. And so that's a positive feedback situation. That's kind of a dangerous thing for the potentially dangerous thing for the nervous system, because if this guy is excited by the colliculus at that location, and then it goes back to the same location in the colliculus and excites that location in the colliculus, well, it could sort of get out of hand, right? Because these guys, you excite, this guy comes down excited, goes more and more and more, you get like sort of epileptic fit, though you get this extreme driving of neurons. And so I originally discovered a completely different connection pattern in another nucleus isthmi, and this one is the GABAergic. So that means inhibitory nucleus isthmi. So there's another one. In birds, it's very clear. You can see there's two very distinct nuclei, and if you stain them, you can see that the one of them is GABAergic and the other one is cholinergic. So what do the cells here look like? The cells are bigger. So the cells in this nucleus are bigger. But more importantly, they actually seem to project either in turtles, they sort of project to the whole of cholinergic nucleus isthmi. In birds, they sort of seem to project more to lines, but they come up into the colliculus and they kind of project across the colliculus in a line. But in any case, and they're synapsing the whole way, in any case, they hit a lot of neurons. So they hit a lot of neurons. a lot more neurons than the terminals like paint brushes and brooms. So they hit neurons all over the colliculus. So, and those guys are inhibitory. Now, what do they get input from? They get input from these cells in the superior colliculus as well. And so this input is an excitatory input. So it's a plus. That's a plus there. But the output of this nucleus is inhibitory. So this is a minus here, and a minus here. So what can this kind of network do? So this kind of network can basically do a winner-take-all kind of computation. And so we talked about that when we talked about recurrent networks. So winner-take-all. So if everybody inhibits everybody, that's basically an example of a connection pattern that's a symmetric connection pattern, and it has stable states. And what are the stable states when everybody inhibits everybody? The stable states are only one guy wins. And so the way this probably works, because you can just guess by just actually, without even recording, just looking at the architecture of it, is that a target comes in. So say a target comes in, like there's you're a frog, and there's a little fly over here, and then there's, say, a worm over here. And the worm is sort of more interesting. And so that will stimulate the retinotopic superficial parts of the superior colliculus, and then that's going to cause some activity to go down to the cholinergic nucleus isthmii, and that's going to come back and stimulate the same place and start a little fire in this part of the colliculus, start a little continuous firing. So even if the worm just moves a little bit, this thing will, like the worm just goes brip, a little movement like that, but that will start off this little firing in this feedback circuit. But as that happens, that starts to stimulate the GABAergic nucleus isthmii, and that inhibits everybody. Or, at least lines. In turtles, it inhibits everybody. It inhibits everybody in the colliculus and everybody in the cholinergic nucleus isthmii. And what is the end result of that? The end result of that, one of the end results of that is that only one guy wins. So who's ever the most active wins, because they cause everybody else to be inhibited. They cause themselves to be inhibited too, but say the moth wasn't quite as attractive as the worm, then there's going to be more activity in this circuit, and then you're going to have this guy win. And so what will happen is you can make a decision. Let's just look at this guy. So we'll suppress the other targets, and then we'll just look at that guy, and so that whatever is the most attractive target at one point in time is the one that you'll make a saccade to. And so we need some kind of decision making. The same thing has to happen in the motor system. If you're grabbing, you've got two things in front of you, and I say I want to grab the yellow one. I don't want to just sort of grab in between. I'm going to say, like, I've made a decision. There's two interesting things there. I'm going to grab the yellow one and ignore this other one completely. So we need this kind of winner-take-all thing a lot. But think about it. Not everybody. So what do I mean by that? Now remember what we said about the colliculus was that it's updating a map of salient targets. So we can't completely suppress all the neurons in the colliculus. So this circuitry that decides we're just going to look at this one now can't wipe out all the activity in the other targets, because we need to update them. We need to sort of move them to a new location in the colliculus after we looked at the most interesting target. So there's got to be a reservoir of other neurons in the colliculus that don't get shut down by this system, and that has to do with what neurons do these things contact. And I didn't really say what neurons they're contacting here, and it's not even completely known what neurons they're contacting. So there's 50 types, right? So you could imagine there could be sort of some layers in there that will maintain the targets, and then other layers that will be sort of more winner-take-all type layers that will decide, like, okay, I'm going to erase all the rest of the targets and just concentrate on the one. So you can see it's a subtle thing. Again, it's not really completely worked out how this all works, but these are just some ideas about how it might work. So that this idea of spatio-temporal to temporo-spatial, and then this winner-take-all idea of like how do we choose targets, but we can't erase everybody because we need to update the other targets. If there's two interesting targets, we look at one, but we want to maintain where the other one was, somewhere in the colliculus. Okay, so that's some ideas for how some of this circuitry, not very well understood circuitry, might work. I mean, it's understood to the extent that we know that it's doing what we said it was doing. It's doing these spatio-temporal and temporo-spatial transformations, then it's doing a winner-take-all because only one saccade comes out of the colliculus when there's multiple targets. But how the whole thing works, given that we've got 20 or 50 or 60 types of cells in the colliculus, is less clear. So go to the MRI center and check out the cholinergic nucleus isthmii. That's the one that gets really hyperactivated when turtles drink coffee, because you can zip up your cholinergic system a little bit by drinking coffee or smoking cigarettes. Okay. So now let's talk about human VIP. So we're going to completely change topics and talk about human VIP. So if we look at monkey VIP, so most of these experiments were originally done in macaque monkeys, and we have to sort of list some of the sulci. So we've got the central sulcus to sort of key off of, and then we have the superior temporal sulcus, and the superior temporal sulcus kind of goes up like that. That's the STS. So that sort of like orients you. Now, if you look at the frontal cortex of a monkey, you've got the principal sulcus and the arcuate, something like that. And then behind there, you've got the intraparietal, and then there's the lunate and the inferior occipital sulcus. But let's just concentrate on the intraparietal. So this guy is the intraparietal. And if we kind of like cut a section down through the intraparietal sulcus to sort of see why it's called VIP. So if I cut a section down and look at it, what it looks like is here's the midline. That's like right at the midline, and then there's a gyrus, and then it goes down into a sulcus like that, and then it comes back up again. And so that would be the intraparietal sulcus there. And here's the other hemisphere, like that. So that's the midline. So where is VIP? So VIP is sort of sitting at the very, very bottom. So this is, VIP. And then LIP is called LIP because it's on the lateral side of the sulcus. So that's LIP. Lateral interparietal, that's ventral interparietal, sort of like meaning down. Now, so I did some experiments, back in the day trying to find lateral interparietal sulcus. And if you look in humans, so here's a bigger human brain. And there's like the frontal operculum kind of crinkles up. There, it goes into the insula. So the sulci in humans are a little different. So here's the here's the central sulcus. And there tend to be sort of like two main sulci up here. It's kind of like you attached up the principal sulcus to the upper bank of the arcuate. And if you go behind the central sulcus, so this is central, you see that there's something called the post-central sulcus. And there tends to be a sulcus sort of parallel to it, and then it stops, and then there's another sulcus. It varies in different people. Like I always say every year, some more monkey-like people like me have a monkey-like intraparietal that kind of arcs around just like the monkey one does. But other people have sort of a post-central, and then intraparietal. And the intraparietal, it's almost on the midline. It's almost on the midline. And so when we first went looking for LIP, they basically sort of found it virtually on the midline, like about there. This is our LIP. And when we were first trying to get this published, the people said, it can't be LIP, it's way too medial. So why is it medial? And so it's important to sort of, when you're comparing species, to try to get some idea of like how things have changed and how things have stayed the same. So if you look at a human, there's an STS, and it often sort of breaks up into a couple. Sometimes people have a STS that goes up, sometimes it goes down, sometimes these two little branches are attached to each other. But if you look at the end of the STS, at the end of the upper part of the STS, you have what's called the angular gyrus. And so it's not a particularly... So what is the angular gyrus? The angular gyrus is basically, end of the STS. And it turns out when people have done these experiments with a resting state, where you just have somebody sit in the magnet and just kind of daydream, you find there's an area here called, the angular. This guy is the angular gyrus. Default mode network component. Is that enough? Very Germanic. So the angular gyrus default mode network component. And so that thing, basically, if you just see what areas are correlated with each other, if that one is on, this guy's hooked up to the hippocampus. So if that guy is on, areas like LIP will be off. And if you're doing a task, like you're concentrating on a little dot that Marty said you have to look at, well, then the angular gyrus will turn off. And so you can just see this if you're just sitting daydreaming in the magnet every once in a while. Oh, I'm stuck in the magnet. I just looked at the screen. Yep, it's the same screen I was looking at before. And then you're sort of daydreaming and thinking about lunch. And so you can see these sets of things kind of anti-correlated with each other. So what did that do? That essentially sort of pushed, it pushed all the areas. It's bigger than it is in macaque monkeys. You have the same thing in a macaque monkey. It's just a little thing up here. It's still there. It has the same sort of resting state features. So that's approximately where the angular gyrus of a monkey is. The default area of the monkey is. It's actually down in the sulcus. So, angular, we'll put gyrus in quotes. Gyrus, because it's actually a sulcus. And so what's happened in humans is it's kind of pushed everything around a little bit, because it got quite a bit bigger in humans. And so that kind of explains why LIP is so medial, because it sort of got pushed out of the way. But if you look at VIP is, if you look at it on a macaque monkey, if you sort of like follow from V1, you first run into LIP, then you run into VIP. So VIP is kind of more forward. And so what happens with VIP in humans is it's kind of sitting here. So that's where VIP is in humans. It's kind of, so it doesn't work with the name, because lateral intraparietal is supposed to be lateral, and ventral intraparietal is not quite as lateral, but here's the other way around. lateral interparietal is medial, and ventral interparietal is lateral. But we just keep the same names to sort of, protect the innocent and try to sort of keep things relatively straightforward. And so what are the experiments that we did? So the first thing we know from the monkey experiments that there's a retinotopic map in here. So let's look for retinotopic mapping. And how do we do that? We just do something like put a wedge on with cows and faces and checkerboards and a lot of things, and then move that wedge, and then what happens is, if we've got a receptive field over here, what's going to happen is we'll get a periodic signal every time that wedge goes around. So the signal will look like, it'll go up and down like that. And then if we measure the phase of that signal, which is kind of the single cycle offset, so here's the phase of that signal, phase. Let's just put the word phase there. It's better. So this guy is the phase. That will tell us it's going to be sort of proportional to the angle, the visual angle, visual polar angle. And so that's how we can sort of map the polar angle of these things. And so to do this, you have to get really sort of in your face, because VIP actually it's kind of concerned about things that are close to your face. And so if things are kind of far away, like a couple feet away, it doesn't care. And so the way this was done was you put somebody in the magnet. Here they are. My regular ugly person in the magnet, and put a screen really close to them, right in front of their face, project onto that screen, and they look up at that screen without a mirror, and so it's really close. So you do that, you find a visual map, but we know that somatosensory, and this is work that I did years ago with Rui-Song Wang, my student. So if you look at the monkey data, we know that there's a somatosensory map in there. So, face, somato. And so we did some experiments. The very first experiment we did was I got a chopstick and I went in the magnet and I just touched different parts of my face as best as I could without moving anything else. I just got this chopstick and just slowly touched different parts of my face. And sure enough, there was enough of a signal there, and so we made a little air puffer that would just allow us to put air puffs at different parts of, so here's a little air puffer. It comes out and it's got these little snake-like things that go to different parts of the face so that we can sort of stimulate around the face. So that's air puffs. It just feels like we don't puff in the eye, so it feels like just a little cool touch on some part of your face. And sure enough, there was a map in there. So how does that work? Well, here's the smiling person in the magnet. And we basically just stimulate in sequence all these different parts of their face. Stay away from the eyes. So you sort of go like this and just go around their face. Similar to the way that we are going around the visual field. Do the same thing, analyze it the same way, find out there's a face map in there too. So basically, we have a retinotopic map, we have a face map. But how could we actually figure out what's going on with this face-centered business? Because we said that this is a special kind of map in VIP, and what happens is if you move your eyes around, the map stays stuck to your face. And so that means you've got to move the retinotopic information around to respect the fact that we're interested in where a visual thing is with respect to your face. So So what we really want is 3. So we want, face-centered visual mapping. So we are interested in the visual signal, but we're interested in somehow sort of referring it to the face. And so how would we do this? Well, so here's our sort of coordinate system on the face. Here's our, visual field coordinate system. So what we want to do is put visual things in different parts of the face. But there's a visual map there too, so we somehow have to sort of like get rid of this visual map. And the way we do that is we just had the person view a video that was in an aperture like this and fixate the center of the video. So we would put a video there, I think we used Xena the Warrior Princess, and so you just watch Xena the Warrior Princess there as this, that sort of dates me, as this thing goes around, so the person isn't fixating the center, the person's fixating the center of the video, and as they fixate the center of the video, they more or less see the same thing retinotopically, because they fixate the center of the video. So fixate center moving video. So that keeps the retinotopic stimulation about the same, because you're always looking at this video and there's a circle of video. But what's changing? So what's changing is where the visual thing is with respect to your face. And so at one point, you're fixating the center of that. There's something sort of near your forehead, then there's something near your left forehead, then there's something near your cheek, then there's something near your chin. But the retinotopic mapping shouldn't be engaged because we're kind of looking at the same thing. But we're looking at the same thing, but we're looking at the thing that goes to different places on your face. And if you do this same kind of analysis, you find out you get the same map. You get this, the same map. So essentially, that was a kind of a tricky way of trying to see if we could see this face-centered remapping of visual stimuli, because that's what's going on. Because what's coming into the system, into the visual system, is the same thing, the Xena video. But we can find out that this area is taking that constant retinotopic stimulation and moving it around in VIP to the corresponding locations of the face. So so basically, if you compare all of these things, if you compare all of these guys, basically what you see is you get the same map. So you get the same map, all cases. And so that's some evidence that, we actually, in this region, in the post-central sulcus, which is more or less at the boundary between the visual system and the somatosensory system, you have this multisensory map that essentially re-references visual information into somatosensory coordinates, face coordinates. And we know that in fact it's a little bigger than that. It extends all the way to the midline, because there's a representation of the toes over here. So if you air puff the toes, you can see that there's actually, it goes all the way down the body, all the way down to the toes. And what portion of the visual field does that correspond to? The very far periphery, low periphery. And so when you're walking ahead, when you're walking straight forward, you can see your feet and you can see what's on the ground. If you block if you're try walking downstairs blocking with something blocking in front of you, you can feel it's not quite right. And that's because you're actually seeing in your far periphery your toes. Okay, so these were fMRI experiments. If you want to look them up, one of the papers is Serino and Huang, 2006. So it's just basically copying the monkey experiments. Okay, Stan, I think we're ready to go. I just